Mitosis versus Meiosis by Nikki, Gwen, and Margot. So before we get into the specific details, let's start off with the basics. The cell cycle. For both meiosis and mitosis, they begin with interphase. For both of them, it's the same process consisting of three main phases, G1, S, and G2. In G1, the cell grows as it prepares for DNA replication. Here it also goes through a checkpoint. Cell checkpoints are little stop signs in the cell that monitor the cell and make sure it's doing what it's supposed to. The next phase is S. Here, DNA is copied. Sweet and simple. Before the cell can begin division, it goes through one more growth in G2, where it also has another checkpoint. So, mitosis. My toes itch. That's how you'll remember it. So, hopefully your toes don't itch, but if you do, you might want to get that checked out. And when they check it out, you're going to find that your doctor is going to tell you that they have to cut it off. And that's where mitosis comes in, as it works with tissue growth. In mitosis, it produces two new daughter cells that are identical to the parent, including the same number and kind of chromosomes. The 6 main stages of mitosis include prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. In prophase, the nuclear envelope disappears as chromatids become more tightly coiled. Spindle fibers also form in the cytoplasm. They're used in another step. Get it? Step with your feet, and your feet have toes. Ha! Okay. Next is the middle in-betweeny phase, also known as prometaphase. The chromosomes begin to line in the middle as the spindle fibers attach to the centromere, the thing holding the two chromatids together. Metaphase is next. In metaphase, the chromosomes meet in the middle of the cell, aka the equator. Meanwhile, the centrals have skedaddled their way to opposite poles. Anaphase is when the chromosomes are pulled by the spindle fibers / microtubules as the kinetochores bite spindle fibers and bring the chromosomes to opposite poles. The kinetochores don't actually bite the spindle fibers, but this is just to give you a visual. The last stage in PMAT is telophase. Here, the nuclear envelope starts to reform around the new cluster of chromosomes as chromatids become less condensed. Here you can see the beginning of 2 new daughter cells. The place where cells are still connected is called the cleavage. Cleavage. Cytokinesis is the final division as you have 2 separate, identical daughter cells. And that's mitosis. Next, we have meiosis. Now, meiosis is very similar to mitosis, but at the same time very different. Interphase in meiosis is the same as mitosis. The cell grows and replicates DNA. Then, in mitosis, you enter PMAT, which is where the cell divides. In meiosis, you still have PMAT, but it goes through it twice. In meiosis 1, you begin with prophase, where crossing over occurs, which helps with genetic variation. The homologous chromosomes line up next to each other at the equator, where spindle fibers pull the chromosomes to opposite poles in metaphase and anaphase. You're left with 2 haploid daughter cells with 46 chromosomes, and that's at the end of meiosis 1. Then it enters meiosis 2. Now, meiosis 2 skips interphase because it doesn't need to replicate its DNA anymore. Meiosis 2 is very similar to mitosis at this point. You have your chromosomes now from your 2 separate cells. They go into prophase, where the nuclear envelope disappears again if it was ever reformed. They line up in the middle during metaphase. The sister chromatids are pulled apart in anaphase and pulled by the spindle fibers, and then you're left with 4 haploid unique daughter cells with 23 chromosomes in each. These 4 new cells are called gametes, which are your egg cells and sperm cells used in sexual reproduction. Wow. Cleavage and toast in the same cell PowerPoint. You don't see that every day. Well, until next quarter, it's been Gwen, Margot, and Nikki. Bye!